So hi guys, welcome to lecture 3 of the DNA, proteins, and their applications biochemistry course. Today we're going to talk about cellular mechanisms of cancer pathogenesis. So, why does cancer occur? Well, cancer is intrinsically a disease of the cell cycle. So basically the cell cycle has been dysregulated, and this dysregulation of the cell cycle happens through subsequent mutations, and in order for cancer to actually occur, there needs to be multiple of these mutations. So let me pull up your guys' worksheet. There we go. So just to go through the cell cycle again, there are two growth phases, G1 and G2, and an S phase. An S phase is where DNA is replicated. So basically what happens is the cell grows, and then it gets big enough and it's like, hey, let's divide. But in order to divide, we first need to replicate our DNA. So then they replicate the DNA, and then they grow more, and then there's signals for the cell to divide, and it undergoes mitosis. And then once one cell splits into two, those two cells now are in G1 again, and they grow till they're ready to synthesize their DNA, and they do it all over again. There's a fancy little arrow right here that says G0. So G0 basically means like the cell is not growing anymore. And there's certain cell types that can enter and exit G0 phase as they please. So an example of this is like your neurons. So I don't know if you guys have ever heard the myth or the idea that, like, you know, once you kill your brain cells, you kill your brain cells. And that's because your brain cells don't really divide after they're done being formed. They kind of they go into G0 phase, which is where they normally just like are cells, and they do their processes, but they're not trying to grow and divide. And there's some signals that can switch a cell from G0 back to G1 so it will replicate, but that's a very tightly regulated process. But overall, there's two phases of the cell cycle. There's interphase, which composes of G1, S, and G2, and then there's M phase, which composes of cell division, which is mitosis and cytokinesis. So on that note, I want to iterate that mitosis and cell division are different. Cell division refers to the process of mitosis and cytokinesis, which cytokinesis is when the cytosols of the cell that's dividing, when they get split. So like all the organelles, everything in the cytosol gets distributed evenly and splits. Whereas mitosis is all about the DNA. So like properly aligning the DNA, or the DNA properly condensing in prophase. And then in metaphase, they line up or they get their spindle fibers. And then in anaphase, they line up at the anaphase plate. Then in telophase, they get ripped apart. But all of this, what we're talking about is we're talking about the DNA. And then in cytokinesis, we're talking about the whole rest of the cell. So all the mitochondria, the Golgi, the ribosomes, lysosomes, everything else needs to get split from the parent cell to the two daughter cells. And so that's the difference between cytokinesis or mitosis and cell division. But I mentioned that there are signals here that basically tell the cell to transition to G2 to M phase. What are those signals? So those signals are called mitogens. And so basically, once cells pass this G2 checkpoint where they make sure cell size is appropriate and the DNA was repaired appropriately, there'll be an influx of mitogens that mitogens are just proteins that stimulate mitosis, is essentially what they are. But a cell cycle checkpoint is like certain proteins communicating with other proteins to be like, hey, did you do this job? And if the other protein's like, yeah, we did this job, and then the checkpoint protein will be like, okay, I'm gonna turn you off now since you did your job and you did everything you're supposed to do, and we're gonna move on to the next phase of the cell cycle. And the reason why I say, oh, I'm gonna turn you on or off is because these checkpoint proteins are all kinases or phosphatases. So kinases are proteins that add phosphate groups onto proteins, and phosphatases are proteins that take off phosphate groups from proteins. And the addition or subtraction of phosphate groups from proteins is a very special event because this phosphorylation event is very high in energy. So a lot of times it can activate a protein. And when you dephosphorylate something, a lot of times that inactivates a protein because you're taking away that energy that's coupled to that phosphorylation. So, like, for the G2 checkpoint, which happens right before mitosis, for example, ATM, which is a kinase, will turn on proteins that stimulate mitosis, and check 2 will turn off proteins that stimulate growth. Because during the M phase, they're not growing at that point. They're doing cell division, right? And so we want to turn those things on that are about to happen, mitosis, that are going to stimulate mitosis, and we want to turn off things that are not happening anymore, such as growing. Or what we call growing in this context, we call it proliferation. But there are 5 crucial checkpoints. So there's the spindle assembly checkpoint, which happens mid-mitosis, basically makes sure that all the spindle fibers have attached to the chromosomes and they're now lining up at the anaphase plate. There's the G0 checkpoint. This checkpoint is kind of like the fake 5th checkpoint because not every cell has this checkpoint, only those that are in G0 or have the potential to go into G0. But the main 4 are the spindle assembly checkpoint, the G1 checkpoint. This basically says, like, are we big enough to replicate our DNA? Yes. The intra-S checkpoint is asking, it's asking, are we replicating our DNA properly? And then there's not another checkpoint until the G2 checkpoint, which is, did we replicate our DNA properly? And is it free of mutations? And are we big enough to divide? So, why does cancer occur? Like I said, these mitogens. So, mitogens can enter all throughout the cell cycle, but they are mostly abundant in the phases leading up to mitosis. Because you can see in the M phase and going into G1, there's not really any mitogens in this area of the circle, right? And that's because all these signals are getting ready to prepare the cell for mitosis, and then once it's in mitosis, it doesn't need any more mitogens. It's already going through mitosis. But an example of how cancer cells are essentially a dysregulation of the cell cycle is normal cells. If you give them these mitogens, the cells will divide. But if you don't give them the mitogens, then you'll get no cell division. But in cancer cells, it doesn't matter if you give them the mitogens or you don't, the cells will divide anyways. And that's because this regulation of the mitogens is dysregulated. So like potentially you have a mutation in CDK4/6, right? And so CDK4/6 helps with this G1 checkpoint. And so if you have a mutation and it's like supposed to activate something for G1 because it's a kinase, and say the mutation makes it hyperactive, it's always going to be activating it. And so it's always going to be pushing the cell forward to go through the cell cycle. But one common misconception about cancer is that you get one mutation and then all of a sudden you have cancer. No, so that's not how it works. You have to have a series of mutations to lead to cancer. So let's look at this hypothetical series. So first you get an initial mutation that inactivates a negative regulator of the cell cycle. So a negative regulator of the cycle is something that turns off the cell cycle. So if you're inactivating something, or not turn off, but should I say, it does not stimulate proliferation. So like a negative regulator is going to be like, don't grow, don't divide, don't do those things. And when you inactivate that, that means you're telling the cell to grow and divide. But because it's just inactivated, these signals of telling the cell to grow and divide aren't perfectly timed. They're always on. And so that means the cell's always going to be growing and dividing. The next thing is you get a mutation that overactivates a positive cell cycle regulator. So a positive cell cycle regulator, we said a -1 basically tells the cell cycle to chill, like don't divide, don't grow. A positive cell cycle regulator tells them to grow and to divide. So if that is overactive, you're going to get even more cell cycle dysregulation of this cycle always being on and having no points of it being off. And then you have a third mutation that inactivates a genome stability factor. So this is something that's going to cause extra mutations in the S phase because the genome is more unstable. And then once the genome is unstable, additional mutations accumulate rapidly, and then you have a cancer cell. So next slide. Oh, so I wanted to say a couple more things about mitogens. That's on your worksheet. So are mitogens or cyclins, cyclins just like a more specific term for mitogens, or it's a class of mitogens. Are they constitutively active throughout the cell cycle? No, because I already mentioned that there's this part of the cell cycle where there's no mitogens and stuff like that, right? Because it doesn't need it, because it doesn't need to stimulate mitosis if you're in mitosis. But furthermore, there are different cyclins and mitogens that peak and fall in terms of how abundant they are throughout the cell cycle. So they're not constitutively on, they're kind of on and off, and they're dependent on which other ones are on and which other ones are off, because they're all like communicating with each other. So one thing I won't go into detail today, but Another really important thing to the development of a cancer tumor is the tumor microenvironment. So this is things like, is there a blood vessel nearby? Because if there is, then there can be intravasation into the bloodstream, and that's how you get metastasis. Is there a rich collagen matrix, or is the collagen matrix maybe not as rich and there's more space for the tumor to grow? What's the hypoxia environment like? Is there bone marrow derived cells nearby? There are many things that contribute to the microenvironment of a tumor. So, your main takeaway from this should be that cancer is essentially a disease of the dysregulation of the cell cycle, and that this dysregulation of the cell cycle stems from mutations in important regulatory proteins that regulate the cell cycle. So, theoretically, we should be able to cure cancer by fixing the mutations in said regulatory proteins. So, now technology is making that theoretical cure a reality through CRISPR-Cas technology, and that's what we're going to go into next week. So now we're going to discuss our journal club, but I'm going to stop the recording.